Welcome, everyone. My name is Kurt Losert. I'm the CEO of the Fibrolamellar Cancer Foundation. And welcome to the second of our four-part webinar series on precision cancer medicine. Tonight, we've got a phenomenal panel for you all to really explore the application of functional precision medicine to rare cancers. This is a strategy where live tumor cells from people with a cancer are directly treated by drugs to provide immediate personalized information to guide the therapy. And it's something that's becoming increasingly important in the clinical community today. So hopefully you'll enjoy this 1 hour webinar. You'll notice at the bottom of the screen in Zoom webinar format, there's a button named Q&A. If any questions you have that come up during the course of this discussion, please type them in there. And our moderator and panelists will all be able to view them and hopefully get you some good, relevant answers to your questions. With that, I'd like to turn the attention over to Mark Firth. Mark is the Director of Research for FCF, and he'll be the moderator of tonight's panel discussion, and he'll also introduce our exciting set of panelists. So with that, I'll turn it over to Mark.
Kurt, thanks very much. Welcome to everybody on our panel and to those of you who've joined in to this. And again, as Kurt said, please feel free as questions arise to put them in that Q&A box because we'll try to address those over the next hour or so. We were hoping to have a panel of 4 really terrific members. Unfortunately, Taryn Gujral from the Fred Hutch Cancer Center is unable to join us because of a case of COVID in his family. But we're very happy to have 3 people who are really expert in the area of what we're calling functional precision medicine, specifically for rare cancers, but certainly they work on both rare and common cancers. The area, as Kurt indicated, includes asking the question, can you study something in an individual patient's tumor in how it actually responds to therapy to predict how to treat that patient? But actually, it's certainly even a broader area than that. In our first webinar, we talked about genomics, basically the study of DNA in tumors and mutations in that DNA that tell us something about an individual cancer patient's tumor and what that might imply for diagnosis and treatment. Tonight, we're going to talk about a whole bunch more of omics, and one can rattle off the names and it becomes almost an alphabet soup, but we'll probably use some of the words. But we're really looking at a whole range of aspects of cell function, starting not with the gene sequence, but what genes are expressed in a tumor cell, how those compare to normal cells, whether they are constituting a pattern ultimately of what determines the physiology of the cell that's distinguished from normal cells in a way that allows us to attack them therapeutically. We're talking about the same question for proteins, now more and more questions also not about genes or RNA transcripts, but also the organization of genes in a 3-dimensional way in the cell nucleus, packaged up biochemically that determine activity of genes. And increasingly also we're looking at metabolomes, as they're called, or the pattern of metabolites expressed by cancer cells and how their overall metabolism differs from that of normal cells. So that we're really talking about every aspect of cell physiology, always with the question, what does that do to give us handles on how to treat a cancer and to find ways to selectively kill cancer cells and do the minimal damage to normal cells in the body? And we're going to ask each of our panelists to talk about how they approach that question. Praveen Setupathy from Cornell University is, of the 3 panelists, the one who is most basic in his approach. He's a PhD scientist, tremendously interested in what we call translation. That's the taking of what we learn in the most basic laboratory studies and bringing it to the patient's bedside. And he'll talk about, I think, the big picture of some of those different omics or omics technologies. We've asked Bob Nagorny, who's the head of the Nagorny Cancer Institute in California, and has been for a very long period of time relative to the field, an expert in actually asking, can one query individual patients' tumors to find out very directly by treating those cells in culture what might work in those individuals as patients. And then Eitan Rupin, who is the head of cancer informatics for the National Cancer Institute, and an expert both on the global integration of the information that comes out in these kinds of studies and in actually choosing which studies are most valuable, and helping also very directly in some cases to use that total constellation of information to predict and help guide treatment of cancer patients. So without further ado, let me ask each of the panelists to talk for a couple of minutes about what they do, how it fits into this big picture, a little more about who they are, and then we'll have, I hope, a very organic discussion amongst the four of us. And again, looking at the questions you folks in the audience put up. So Praveen, maybe you could start by talking about that omics universe and how it actually fits into understanding a cancer in a way that gets us to ultimately individualized treatment.
Thank you very much, Mark, and thank you, Kurt and Mark, and to all of those at the FCF for running this series of events. It's really fantastic, and I'm honored to be a part of one of these. I thought I might start actually by talking about what precision medicine means to me as someone who is engaged in a lot of this omics that Mark just mentioned. And an analogy I always find is very helpful, and I hope you'll find it useful too. Imagine that the question that we're trying to ask is, how different are all of our faces on this webinar? But all we did was to take a small section, a dot on each of our foreheads, and compared it. It looked pretty similar. Maybe there are some subtle differences, but it would look really, really similar, right? If we had access to our entire portrait, to our entire face, our visage, then we may be able to see all kinds of differences, right? The way that our eyebrows are shaped, our eye color, whether we have beards or not. Looking at the big picture allows us to see what some of the unique differences are for each of us that are on this webinar. That's kind of the way that I think about the way that omics helps with precision medicine, right? We have 20,000 or so, depending on how you count, genes that are encoded in the human genome. If we hone in on just one or two of those, it might be hard to tell how different we are or how different some of our tumors are from one another's. But if we're able to look at all 22,000 of them all together and how they're behaving in relation to one another, right, then we're able to build a more rich and comprehensive picture, kind of like looking at the entire face. We often refer to that actually as a molecular portrait of a tumor cell, much like we might take a picture and look at our whole face and say that it's a full portrait. We call it a molecular portrait of a tumor, as opposed to just looking at one very specific molecule or aspect of the tumor. The reason why that's really advantageous is because we get the big picture, right? So about 22 years ago, this idea was applied to breast cancer. While most clinicians and researchers were well aware that breast cancer wasn't just one monolithic entity, we really couldn't pin down exactly what different kinds of breast cancers people had. And this was problematic because it seemed that some people had different subtypes of breast cancer, some people responding to some medications better than others. If we had a way of deciding or determining which subtype you were in, we might be able to get you on a treatment regimen that's more effective more quickly, right? So there was a revolutionary work done in the field of being able to do omics, look at all of the genes and their on-off patterns, which genes are on, which genes are off, look at all of them together, and do this across tens to hundreds of different tumors from different patients. And what they realized is that it wasn't just one pattern that was emerging for breast cancer patients, there were at least 4 or 5 different patterns that were emerging. Indeed, every patient had some aspects that were unique, but if you looked at the overall patterns, there were at least 4 or 5. And we now, 2 decades into that kind of research, fully appreciate what those subtypes of breast cancer are, and clinicians can readily diagnose you into one of those subtypes and get you on a medical regimen or a treatment regimen that's going to work much better for you if you're in subtype A versus, say, subtype D, right? This is exactly what we would like to do for the rare cancers that we study, including fibrolamellar carcinoma. The main challenge, of course, is being able to get access to enough samples so that we can begin to build a really big database and a big picture of the variation, the differences that are present in these molecular portraits across many different patients. But as we continue to build and we do the omics to define the molecular portraits of each of these tumors coming from patients, we'll get a better and better picture of what kinds of differences there are. What are the similarities, but what are also some of the differences? And can we take advantage of learning about those differences to come up with tailored approaches for individuals with rare tumors?
Thank you, Praveen. Let's turn to Bob Nagorny and really look at, in some respects, the end result of all of these many differences that one may see from subtype of cancer or just from patient to patient. And again, the effect of querying individuals, patients' tumors very directly to see what drugs they might respond to in the patient. Bob, would you tell us a little more about yourself and that approach?
Yes, thank you first of all for inviting me to join you. I'm so impressed by all the work you're doing, and Praveen, it's been my pleasure to pull a couple of your papers and look at some of the great work you're doing. And Entails, we hadn't met before, I also had a chance to look into some of the work you're doing. So I'm humbled by the opportunity to do this because you're some terrific scientists here. I'm a clinician. I'm a medical oncologist that runs a laboratory. I guess in a way, my work grew out of a desire to improve therapeutics some years ago when things were extremely crude. And as Praveen mentioned, the work of Peru, where they identified new subtypes of breast cancer, and some of the work that's gone on other diseases. You have to understand, some years ago, there was nothing. There was no concept of how tumors differed. And so everybody got everything all the time. And it was just terrible to watch patients suffer through toxicities with response rates in the range of 30 or 40% at best, and very few curative. So I began to be interested in cell biology at sort of a systems level, looking at tissue culture in aggregates of cells isolated directly from patients. And instead of propagating or subculturing or amplifying, I was looking at explants, human tissues directly out of patients without really doing anything to them except separating them into clusters that would allow me to examine what happens when they were stressed, what happens when they were exposed to something injurious. We started off in leukemia and showed rather quickly that the induction of apoptosis, which at the time had not yet really been fully applied in this field, but in the 80s and 90s, as the concept of programmed cell death became more apparent, we began to work on how leukemia patients responded. And then we moved into solid tumors. And it was really at the time of solid tumor work that we realized that cancer wasn't a cell, but a collection of cells talking to each other. Tumor cell to tumor cell, tumor cell to stroma, stroma and tumor cell to vasculature, vasculature to T and B cells, macrophages, cytokines, everything that made cancer as a system deranged. And when we moved from leukemia to solid tumors, a lot of our interesting work seemed to sputter because we had gotten so good at isolating cancer cells that we had isolated them from each other. And there's a well-described phenomenon in cancer biology called anawekas, which translated means death by homelessness. And what it means is that when a cell is separated from its normal environment, it's not happy. So I had to go back and kind of dirty up my cultures. I had to turn them from beautiful single-cell suspensions that look so great under the microscope back to these gamiches of cells in clusters and groupings. And and develop aggregates of the appropriate size to be able to permeate them with these molecules and get signals for cell death. And we began to realize then that apoptosis was only one of many forms of cell death. There's ferroptosis and necroptosis and autophagic, and there's a lot of ways cells die. But as a clinician and sitting in the audience with the people here, the investigators here are of such immense capability, and they have brought such brilliant technologies and capabilities to their studies. I feel a little humbled because I'm just a clinician. I'm really just looking for better ways to treat cancer patients. And I was looking for models that would enable me to take great scientific discoveries and turn them into practical utility. So I'm truly a translational scientist because I'm not a drug developer per se. I do not do very much in the way of molecular biology or transcriptomics or proteomics. I really just do functionality, and I study human tumor function. So my laboratory is studying now human phenotypic response to injury. We've migrated to another level now in metabolism. So we're doing mass spectrometry to examine metabolomics, but we're still very interested in cell function, cell functional response to injury, and ultimately cell death. As many people have said before, the only good cancer cell is a dead cancer cell. So my job is to kill cancer. And I guess that's hopefully what we can do to help these unfortunate people with fibrolamellar cancer.
Yeah. That's a tremendous introduction, Bob. Thank you. And we'll come back to each of the panelists in turn, but I want to turn now to Eitan Rupin, who's, as we said, looking at one level at the very big picture of how to integrate all of these different kinds of data, and also looking at individual patients to try to help predict for one individual in situations where one doesn't always have access to the kind of testing that Dr. Negroni was talking about, but to try to predict the best way to treat individuals from this overall set of information one can get by interrogating, especially the genome's activity. So Eitan, would you tell us a little more about your focus, and maybe now or later we could talk about some specific examples of how you've been able to predict, based on some of this kind of information, a good way to treat a patient, and in some cases had very good outcomes. I think you're still muted. You're still muted, Eitan. Thank you.
Yes, thank you so much, and thanks for the invitation. And I hope the audience is enjoying listening to this discussion as I am. Yeah, so I am an MD, PhD, and my research is devoted to try and analyze large-scale molecular data from tumors in a translational manner in order to try to identify new drug targets for treatment, combinations, and ways to stratify patients to the best treatment. So many of you may have heard about precision oncology and the advent of precision oncology, and in the last 10 years, you may have heard that there is big sequencing companies that are offering sequencing services to patients. And most of that effort is based on sequencing panels of mutations in, say, 400, 500 genes that are considered to be cancer driver genes. Genes, you may have heard the term, that are oncogenic, either oncogene or tumor suppressors, and mutation in these genes are thought to drive cancer, and hence the idea is that targeting them could be a good way for treatment. So these panels in big medical centers are now routines, and people are looking for actionable mutations, okay? Mutations in these cancer driver genes, which the big pharma companies have already devised what we call targeted therapies. These are not general chemotherapies that kill all cells. They're not really selective, and hence may cause serious side effects and all that goes on with chemotherapy. but they are aimed to be more selective, specifically targeting these mutated cancer drivers. And these regimens have shown in specific indications considerable value, but only about 15% of cancer patients have these actionable mutations. So these currently can benefit from such treatments. And many times, regrettably, though the initial response is great, resistance may develop. And in time spans of 1 year, 2 years, 3 years, and so on and so on. So we really would like to be able to do better. And my lab, together with other labs in the world, are looking into other types of omics molecular data beyond the DNA sequencing to try to identify treatment vulnerabilities. And one striking such molecular data that is really, at the tip of our fingers this day is the expression of the genes. As Praveen said, in each tumor or cells and so on, which genes are on and off. And instead of just looking at these driver mutations in 400, 500 genes, we are now able to look via this, what we call RNA sequencing techniques, at the gene expression state of all 20,000 genes. And actually, we can look at RNA beyond the genes and so on and so on. But the challenge is, can we look at this expression and develop methods to actually recommend patients to treatments based on that? And this is the quest that my lab has been working on in the last decade. And we're all the time trying to improve on what we are doing. It's a big challenge. Our prediction accuracy, as we measure it retrospectively, which means on existing data sets, is about 70 to 80%. And of course, the acid test is how well will it work going forward in what we call in medicine prospective studies. So luckily, really with generous support from NCI leadership, we, together with many clinicians, it takes a village. We are pending final ethics, what we call IRB approvals. We are gearing up to start prospective clinical trials in 5 different cancer indications this fall, and test our ability to recommend the best treatments to cancer patients in the NCI Clinical Center, the best as exists, by looking at the combination for each patient of these DNA sequencing, this RNA, the transcriptome sequencing, and another layer, which I will not get into, which is the methylation patterns of the genes. And, it will take us a year or 2 or 3 to assess. But if and that's a big if, should be assessed carefully, if our predictions and expectations are as we think, we hope to be able to increase the number of patients that can benefit from precision-based treatments from 15% to 50%. However, we haven't yet shown that. This is our task and yet to be seen. This is all on common cancers, okay? So we will study specifically advanced aggressive stages of these common cancers, but common cancers like kidney cancer and breast cancer and prostate cancer and so on. The challenge remains what to do with very rare cancers. And they differ because, as pointed before, we have much less data. It's a number game, okay? in order to make interesting statistically significant findings, we need to have large numbers. However, do not despair, because recent technological advancements, which are called single cell-based technologies, are now enabling us to create large numbers of samples, even in rare tumors. And the hope is, and this is a process that superb labs in the NCI are working on, and my lab is collaborating and trying to help, where we are learning how to develop these methods to develop approaches for precision oncology, which are single cell based and hopefully would apply to rare tumors. As usual, one step at a time. I cannot yet promise, but only thing is I can say is that it's central on our map, and we are trying to work hard on improving these methods. So,
Eitan, thank you. That's
yeah.
great. Eitan's already introduced really how some of this technology specifically applies to rare cancers. I'm going to ask Praveen to amplify on that in a slightly different way. We've heard from Eitan about this notion that one can create targeted drugs that work in a different, more precise way, and hopefully carry fewer of the broad kinds of side effects that have been associated with the more general chemotherapy approach that's been a dominant one in cancer, going back 60 years or so now. And we know that those kinds of drugs, more generally, are very general killing drugs for rapidly growing cells or cells that are dividing. And there's a fair amount of collateral damage to normal cells because we have healthy cells in the body that also have to continue to make DNA and replicate and divide. So that's been a big issue in chemotherapy all along. Praveen, when you talk about precision medicine for a very rare cancer like fibrolamellar, sometimes those are also more homogeneous than a big lump category like breast cancer or lung cancer. In the case of fibrolamellar, the one that we think about as a foundation most of the time, there's one so-called driver gene which is found in close to 100% of the cancers. Nonetheless, there's variation from patient to patient, and sometimes it's hard to actually develop an appropriate drug for what looks like it might be the best driver. As we talked about in the first webinar, if you succeed, as was true in a disease, leukemia called CML, chronic myeloid leukemia, that's sort of the poster child where a targeted drug really changed what had been a lethal disease to one where people could survive and to a much longer lifespan and die of something other than the leukemia. But as we've heard, that's not been the general pattern with the targeted drugs. They've not always lasted as long. Nonetheless, when you have a cancer, Praveen, it's a rare cancer, even when it's more homogeneous, and you can't actually go right after the driver, what are some of the strategies to use all these techniques to understand other ways in which you might intervene and still selectively try to kill the cancer cell without as much of that collateral damage as we see with broad chemotherapy?
Yeah, that's a great question, Mark, and it's something we think about a lot. And yeah, it'd be ideal to be able to go after what we see as the main driver gene in fibrolamellar, for example. But we can't put all our eggs in that basket because it might turn out to be more challenging than we anticipate. There may be more obstacles to overcome there. So one of the things we think about is what is the driver protein really doing? What's immediately downstream of it? And what are some of the key players downstream that are kind of the generals, if you will, that are executing the orders of this driver protein? And could we go after any of those? Might some of those generals, those downstream molecules that are really getting the job done of what this driver protein wants to do, could those be more targetable by therapies? And we refer to those as targeted molecular therapies. We're not going after a general phenotype, but we're going after a specific molecule that we think might be really critical in the cancer. that sounds appealing, but how do we identify those? Which molecules are the generals of this main driver protein? How might we go about detecting those? One of the strategies that we use is that in the DNA sequence and in the packaging that DNA is sort of wrapped within in the nucleus of all of our cells, cancer cells have a tendency to make special marks right next to those genes or molecules that are really important for its survival and for meeting its special energy demands. Word was used earlier called an oncogene, a gene that perhaps is particularly critical for the maintenance of the tumor cell's desire to be highly proliferative and active and increase in its ability to move around, invade other tissues, and metastasize. These marks are something that can be deposited right where in the genome, right next to these genes that are really critical for the ability of the tumor cell to carry out its nefarious behaviors. And what we can do using omics is to screen the entire genome of tumor cells that we're able to acquire from patient biopsies and look to see where those special marks are. And we can, if we can look at this in as many patient samples as possible, we can gain some confidence if we begin to see those special marks, the cancer cell having deposited at the same places over and over again, irrespective of whose patient's biopsy we're looking at. And that allows us to then determine that the genes that are nearby might be particularly special, particularly critical for the tumor to be able to carry out its functions. These might be the generals, if you will, that are immediately downstream executing the orders of the driver protein. One or some of those might be particularly targetable. What we've done, we've taken an approach like this in fibrolamellar, and we've come across a few cases of genes that have these really special marks that appear to be critical for the tumor cell to be able to survive. And the really attractive thing about these genes as potential therapeutic targets is that they're not normally active in healthy liver cells. So if we were to be able to develop a drug that targets these molecules, we could deliver them to the tumor and actually not have a lot of worry or concern about what they might be doing in neighboring tumor cells, because those molecules aren't even there. They're being specially activated and used by the tumor cells alone. Right? Those are particularly attractive candidates for us to then work on developing therapies to target very specifically. And just to piggyback really quickly on a really nice comment from Eitan about single cell, instead of doing this with the entire biopsy that we might get from a patient's tissue, we now have the technology, as was already alluded to be able to look at each and every cell in the tumor individually and specially. And why that's really powerful with what I was just saying is it allows us to say not only that we've figured out what one of these generals are, but we can also figure out exactly which kinds of cells is that special gene being activated in by the cancer. And that can be really informative, because if we want to deliver a drug that's targeted to that gene, if we know that it's in a special type of cell, we can maybe try to deliver it to that special type of cell to limit what kind of havoc it might wreak if it went to neighboring cells in the vicinity.
Thanks, Praveen. The studies that Bob Negroni talked about, he actually described moving from trying to work with the purest possible cell population to recreating the neighborhood, if you will. And you know, we always use like that phrase, it takes a neighborhood, or it takes a village, takes whatever, but the village for a cancer cell includes cells that it needs for its survival, like the ones that bring blood to it. It also includes cells that are nominally there potentially to attack the tumor cell. We haven't talked about immune therapy very much yet, but that's certainly the key aspect. And let me ask, Bob, you've developed these systems where you look, either tumor slices or little mini tumors, if you will, in culture. What have you learned from that about going from that individual patient's tumor to the kinds of factors that let you predict an individual response? And how accurately does what you see in that laboratory-based example of the village or the neighborhood in then being able to treat a patient and go after a tumor in that much larger context of the cancer throughout a body? You might want to unmute, Bob. Thank you.
Or coming at this from a clinical standpoint, I guess I would view this as sort of bottom up. I'm listened with great interest to the extraordinary work that people like Eitan are doing and Praveen on, and I guess in a way I would view that as a sort of top-down science, like digging into these cancers through a very bioinformatic approach. I guess my approach is sort of bottom up. I'm kind of looking at without knowing what the driver is, I'm looking for an answer that gives me a clinical information. I remember some years ago when I lived on the East Coast, there was a competition in New York Magazine. They used to ask funny questions, and one of them was for people to make up an aphorism that was meaningless. And one of them was that a stopped clock is right twice a day. Another one was dead fish don't swim home. And so I've come from the dead fish don't swim home school of cancer biology. If I can kill it, I've done my job. And I am so interested and so fascinated by the expertise and the brilliance and capacity of the scientists we're speaking with to delve into why cells die and what's driving this. But as a clinical physician, I really mostly care that they die. So what we've been able to do, and then as I said, this was an iterative process. We started off with these beautiful monocultures, and we went through all these different techniques and growing and propagating and looking at thymidine incorporation. I've done everything. But we finally settled in through working with John Reed years ago and others. We finally realized that cancer didn't grow too much, it dies too little, and if we're going to get anywhere, you got to kill it. So the dead fish don't swim home school was kill the cell in an environment that in some way or other predicts the clinical outcome. And there was a lot of work, it was years of work to migrate from the simple system of leukemias, which die almost uniformly by apoptosis, to solid tumors that die by a variety of ways. And so we began with the study of membrane integrity and the features of cell death at the level of morphology and function. We moved into various biochemical measures, ATP content, mitochondrial function, and now it's a kind of mixture of these. But the ultimate measure of whether it worked or not was whether patients got better. And we started off by taking drugs that we knew about and looking at them in tumors that we knew would respond. So platinum for lung and dexamethasone for leukemia. And then we moved forward in looking at patients who had received these treatments. Could we show in retrospective studies, did the people who we thought would get better? And this is, I think, where Tom was saying they're doing these analyses. And then the final hurdle was, could you start with a patient, test them, and treat them and see a better outcome? And so that's proof for us. We've done that in lung, we've done that in breast, we've done that in ovary, and other diseases, where we can show that when the tissue culture responds for whatever reason, and I'm agnostic as to what it is necessarily that kills the cell. If the cell dies in this model, it is more likely that the patient will respond. We've approximately doubled responses, and we've added to survival. So I guess in a simple way, it works, and we apply it because clinically we're looking for things that work.
Mark, I think you're muted.
We all have to watch that mute button. We've got a question from Dr. Paul Kent, who is well known in the fibrolamellar community as somebody who's put a great deal of energy into that particular rare cancer. And the question is one that actually fits with something I was going to ask. So he points out that for some targeted therapies, for a rare cancer, if you have, particularly if you think there's a good target that's not shared with many other cancers that are much more common, the motivation for people to work on that, and for particularly for biopharmaceutical companies to invest in a project like that, can be market limited. It sounds crass, but that's one of the realities of the world. And so he asked the question about, doesn't it make more sense to try to repurpose drugs that are already available and could be brought to patients very quickly? I can just quickly give one of my own perspectives, which is, of course, the answer to that is yes, if you have the right ones. But it also raises the question, and some of this comes to all of the panelists. We have this wealth of information about a human genome with 20-some thousand genes. We have perhaps a couple of thousand of approved drugs that have gone all the way through in patients, and you can get collections of maybe up to 10 or 15,000 compounds that have been in human beings and at least are drug-like in some of their properties and reasonably safe in human beings. So repurposing is a very broad term, and in general, there's repurposing of cancer drugs, like all the chemotherapy drugs, which can be applied, but that's a relative handful in that universe of 10 or 15,000 compounds. And the kinds of tests one can do if you're looking directly at patient samples, as Dr. Nagorny does, might be what, at most 50 or so for a given patient, well, something in that order, if you're doing really well.
Sorry, a number of drugs we can test, did you say?
Yeah,
it might patient. It might crest out at about 50.
Yeah, 50, drugs or combinations. So there is, even if you're trying to repurpose, a big gap between the universe of what's possible and the universe of what you can test for any one patient. So Eitan, maybe you could help us understand a little bit, how do you bring those two worlds together? At what point do you see this intersection between the huge bodies of data on 20,000 genes and tens of thousands of compounds and huge transcriptome and proteome databases, and really then getting down to what's actionable with existing tools for the patient who's in front of you or one of your colleagues today?
Thank you, and thanks, Paul, for the wonderful raising this wonderful question. I believe we met in the past. So actually, before answering your question, I want to make a comment, and that is that drug repurposing, regrettably, is also problematic from a commercial standpoint. Because many times, even if you find a successful repurposing opportunity, it is not clear how the pharma can make money of it because of different IP and patent issues. The subject is very complex, and that's another hurdle and challenge on its own. But let's assume that's solved, and then of course drug repurposing could be wonderful. So there are two challenges here. First of all, you need to understand better the molecular basis of the disease that you're studying in order to identify potential targets. Okay, so that's a whole challenge of its own, because you can't just throw all the 10,000. I mean, it's beyond our means. So you need to understand potential targets. Once you do that, then you need to rely on some knowledge which of the 10,000 potential drugs is likely to bind and inhibit and be effective to the targets that you found. And what turns out is that most of our drugs are small molecules, And regrettably, the mapping, the knowledge of the targets of small molecules is very noisy. So even if you look at well-accepted databases, those who you know, like Drug Bank and so on, and there are reported small molecule targets and so on, a lot of them are very noisy, to put it gently and mildly. So another big computational challenge which many groups in the world are looking into is to be able to correctly predict the mapping of drugs and small molecules to the targets. So once we know the targets, we can repurpose. Actually, this is a huge project in my lab, which we are now converging and writing up. And as anything we write up these days, we make publicly available in a few days before journals of anything that the audience should know by an amazing service which is called bioRxiv. So we can put our results publicly available way before they will appear in the journals and whatnot. And we do it all the time for anything that we learn and study. I'm very happy to do that. And so in the next few weeks, we will put this new pipeline where we really achieve, I think, to the best of my knowledge, unprecedented accuracy in predicting the targets of 1,500, not 10,000, but 1,500 small molecules, and validating it and so on and so on. So, and other groups are doing parallel wonderful work on that. So that's the two-step process I envision. And I think that in the near future, we will know much better the targets of the small molecules, and that will give us a tremendous leverage in drug repurposing. So I apologize for the long answer for the short question, but right.
That's a very good answer. Thank you. I would be remiss, I think, if as part of this discussion, we didn't talk about some of the other modalities of cancer therapy, some of which, again, are being translated very effectively from first having been introduced in quite common cancers, but now are an important part of what's being done for rare cancers as well. And I refer very specifically to immune therapy. And, you know, there was a dream for decades, going back really to before the Second World War, a recognition that cancer cells were subject to attack by a patient's own immune system, and a dream that one could harness that and amplify that effect to really help a patient to kill his own cancer, if you will, or at least for the immune cells of the patient to do that. And in the last decade or so, that's in fact become an increasingly important part of the armamentarium of oncologists, that there are now drugs, and they fall in a couple of families, as you folks know. One family is, or one approach is to try to be very specific in targeting an individual's tumor, maybe through a vaccination process that's patient-specific. That's one kind of thought. Or in the case of a cancer like fibrolamellar, which again, you know, has a common driver, the question arises: can you attack the driver? But the more general approach these days has been to try to remove blocks to the immune function, and some of that is based on understanding exactly why cancer cells are not killed even when a patient might have mounted an immune response against that cancer. So I'd like to ask some of you to talk about, where does immune therapy fit into this personalized approach, or into this at least, you know, rare cancer specific approach to targeted medicine, or to medicine, to cancer medicine broadly? What are some of the ways that we can use these tools that we've talked about as well, either individual screening all the way up to the global omic kind of analysis, to figure out how best to harness these very powerful new drugs? Bob, would you like to take a first crack at that, maybe?
Sure, Mark. Yes, you're quite right that immune therapy has come into its own in recent years. And after a very long process going back to, I think you were mentioning probably Cooley and Coley's toxin, and the idea of harnessing immunity, One of the questions has been, what is a cold tumor? Why is it that we cannot get responses in pancreas? And it has to do with antigenic accessibility and mutational features of the cells. We know from work done at Johns Hopkins that patients who carry, for example, mismatch repair bristle with targets. And so where the average tumor cell might have 75 neoantigens, someone with mismatch repair, which is a DNA repair abnormality that results in all these protein expressions, they carry 1,100 targets. And so not surprisingly, there was brilliant data reported at Sloan Kettering, 12 patient, 12 responses in the recent ASCO meeting, where patients with mismatch repair responded perfectly to a simple therapy and had complete remissions with an immune therapy with almost no toxicity. Could we do that in this disease? Well, thus far, and I'm sorry, I'm remiss, I don't know the immune status of this disease so well. Maybe I should turn this over to one of the other speakers, because I'm sorry, I don't know that much about fibromyalgia. I didn't think that it was showing a lot of immune targeting, but maybe one of the other, particularly Eitan or Praveen, maybe you know more about the immune status of this disease.
Praveen, do you want to?
Let me respond for 1 second on that, and then I'll pass it on to you because I know something you're going to say. And, you know, what we're saying for fibromyalgia will apply to many other rare cancers as well. But you're correct, Bob. The immune, what people call the tumor mutational burden, which is a fancy way of saying how badly does the DNA in this tumor get messed up, in order, and as a consequence of that, generate lots and lots of triggers for the immune system. So fibromyalgia is at the very low end of having a messed up genome. And so what we've turned also with the same people at Hopkins you were alluding to, actually, is to ask, can we take that one common feature of all fibrolamellar tumors, which is the abnormal fusion protein that drives the cancer, and actually get the immune system to very specifically hone in on that with a vaccine, actually coupled with those immune block removers, the so-called checkpoint inhibitors. And the first clinical study of that has just enrolled its 12th and final patient, and we're keen to see the final result of that. But we already can say at least a lot of the patients are making an immune response. How to get that to really help them medically is still a challenge, although there have been some good responses clinically in that group. But let me turn to Eitan, because there's this more general question. Even with these so-called checkpoint inhibitors, which have revolutionized cancer medicine, especially for the cancers where the mutation level is super high, but even for some of those where it's much lower, how do we actually still face the problem that only a minority of patients respond well? And are there ways we can use the omics world to help us figure out where the blocks are coming and fix it?
It's a $100 billion question, right? So just to tell the lay audience, you know, this immunotherapy endeavor has won some amazing scientists the Nobel Prize in Medicine, and justly so, because its development has been truly excited, because in difference from targeted therapies and so on, immunotherapy, when it works, can really, you know, come the closest to what we can say that we heal the patients. So by now we are seeing patients, even in aggressive states, that were treated with immunotherapy, you know, disease-free for 10 or 20 years. This is, and I'm talking about adult patients. We of course see that in pediatric cancers with other types of treatments, but in adult cancers, until the advent of immunotherapy, we haven't seen that frequently. And that is really transformative and exciting. However, there's two main howevers. One of them, it's mainly in specific cancer types and not in others. Okay, and we don't understand completely why. And my lab and others have published quite a few papers trying to chart the molecular landscapes and explain it and so on and so on. And then we write papers, but we don't really deeply understand that. And the other one is that within a cancer type, even if it's overall responsive, Some patients will respond and others will not, and a lot of effort is invested in trying to stratify the patients. Mark, do you want me, and do we have the time to tell about a specific case report in FLC that somehow I've been obliquely involved?
Yeah, if you're willing, I think it'd be a great thing to do.
Right. Okay, so the story, very briefly, is about a patient, a woman, 36 years old, back in Israel, that had FLC about 3 years ago, 3 and something, and came to Sheba Medical Center, which is one of the best medical centers in Israel, and had a resection of the primary tumor, and the material was sent for molecular fingerprinting, both what we said DNA and RNA, the gene expression and so on. And she didn't have the conventional biomarkers for immunotherapy, okay? The levels of these checkpoints was low, the tumor mutational burden was low, there was no mismatch repair. However, the clinician who treated her, the brilliant clinician Raanan Berger, started her on a specific regimen of a specific immunotherapy with an anti-angiogenic therapy, but she did not respond, regrettably. And 2 years ago, in June 2020, she came back. There was a big relapse of a disease, metastatic and recurrence, and she was hospitalized. And then Raanan Berger turned to a startup in Israel, which, in full disclosure, I have to say I co-founded and I have divested from, and I'm a non-paid advisor, but I still should mention that. And I should not say its name, not to do any commercial or whatever. And they worked together, and that startup developed a technology to look at the sequencing, but instead of just looking on the levels of the direct biomarkers, these checkpoints, it looks at the level of their partners, as Praveen explained, those that work with them together. And by looking at the level of these partners, they developed some way to score which patients are actually likely to respond to immunotherapy, even if you don't see the direct evidence. And in that case, and you know, it's a case study, every person is an island and should be taken carefully. Actually, that scoring predicted that that patient would be highly responsive to a specific combination therapy of anti-PD-1 and CTLA-4 and so on and so on. And she was put on that therapy in June 2020, and by now she is totally disease-free and is doing very well. And gradually the therapy was tapered off. She is now just on anti-PD-1 and so on and so on. There are a few more patients that are being treated with that, and it needs to be seen. But it's really shows you that the need to develop better biomarkers to stratify the patients beyond looking just under the lamp. And perhaps sometimes you get lucky, you know.
Yeah. Thanks very much for sharing that, Eitan. We're close to the end of time. I don't see open questions, so what I'd like to do is just ask each of our panelists to take a minute or so and give us any final thoughts. And as I do that, let me thank each of you. This, for me, has been fascinating and informative, and you know, it's the sort of thing where I'd like to sit down with all of you and continue the discussion until well into the early hours of the morning. But in the interest of our kind audience, Praveen, let me just ask you, you know, we've heard a lot about this, the big approach, the bottom up and the top down, and some of the benefits. Can you help think about ways that we can continue to bring those all together, ultimately to do the best we can by patients, and especially those patients who have the rare diseases which are otherwise tend to be ignored.
Yeah, that's right, Mark. That's really, I think, the holy grail is if we can bring those two approaches together, because I think they have a lot to learn from each other. Both have some limitations. I was responding to Paul's comments, Paul's question on the chat, and one of the things I was saying is sometimes going after the identification of specific targets can actually reveal broad pathways that are aberrant in a tumor that may already have drugs that target it. And so while the initial effort may have been to identify specific targets, it can be revelatory about overall broad pathways or networks that are changed in the tumor that may already have drugs that target it that can be repurposed, or maybe a collection of targets. And so rather than maybe screening 5,000 compounds, perhaps some of the omic and the targeted approaches can actually help suggest that a collection of 100 of those or 50 of those may be most worth investing in for some of the screening studies. So, and then vice versa, as we identify compounds through screening-based approaches that seem to be doing a reasonable job at killing the fish, as Bob would say, we can work backwards to say, why might that be? What are some of the shared features of some of these compounds in terms of the pathways or the molecules that they may be involved in hitting? And can we use that information to develop even more targeted approaches that can be useful for a broader collection of patients? So I think it's the marriage of the two approaches where we're, I think, going to see the most success.
Thanks, Praveen. Bob, could you have any final comments you'd like to offer, please?
Well, I think that the discussion really appropriately has focused in on melding different modalities and melding different disciplines to come to answers. Rare tumors offer an interesting opportunity. It's hard to go wrong with a disease that's so difficult to treat and so hard to find and so hard to study. So it's almost everything is moving forward. Cancer is humbling for its complexity, promiscuity, and redundancy. And one of the problems we have is that we do tend to sometimes be so reductionist that we drill down on something and we go after a target. And luckily, sometimes Brian Drucker was brilliantly successful in identifying a compound that eradicated CML, and everybody wants to do that again. But I think that maintaining this, what I've said before as a clinician is that cancer medicine is a little bit like an ultrasound. The ultrasound sends out waves. You've got to be prepared to receive the echo. You've got to get back what it's saying. And I think what Praveen was saying is, let's not only do things, but let's listen to what the cancer is telling us. I mean, that's a funny thing to say, but I really think tumors are telling us something, and we should be listening. So I use my approach in cellular biology. All of these modalities, I think, will ultimately give us answers. And I'm immensely impressed and delighted to be part of this, Etan's work and Praveen's work. And I'm so sorry we didn't have a chance to talk to Turan, because I was very interested to hear his organotypic approach, but maybe another time. Anyways, I appreciate the opportunity, and I think there's a lot to be done here.
Yeah, I'm reminded a little of Siddhartha Mukherjee's great book, The Emperor of All Maladies, which he calls a biography of cancer, as if we're a living entity, and certainly one that one has to listen to and go beyond one's maybe scientific comfort or medical comfort zone and preconceptions. Etan, let's give you the last word on the top down, bottom up, and the marriage thereof.
I actually want to talk about something else, and that is in an orthogonal dimension.
Hi, did we just freeze? I hope he comes back. That's known as ending not with a bang, but a Zoom whimper. Oh, I'm so sorry, folks. Well, We'll have to bring Eitan back to give us whatever his orthogonal thought was. Kurt, is there anything we can do at this point, or I think we may have to?
I don't think so. It looks like he's
Okay.
lost his internet connection. So,
Yeah.
but with that, unfortunately, Eitan had to leave us the wrong way. But we appreciate everyone's
He may
time.
be back now.
Oh, there he is.
Sorry, I
Wonderful.
probably
That was a great way to build the suspense. Why don't you tell us what you were going to tell us?
right. No. These two disorders, private foundations have changed the disease. So one of them is the case of cystic fibrosis, which huge contributions were made by the relevant private foundation. And the other one is SMA, spinal muscular atrophy, where again, the private foundations played a central role in funding the efforts of brave and brilliant individual scientists who transformed the disease. Are you familiar with the case of SMA? Kurt, Mark, others? So, it's an amazing work of a scientist in Cold Spring Harbor, 25 years, and so on, which was heavily funded by the SMA Foundation and literally single-handed transformed the disease. I recall just before the pandemic, he came to speak at the NIH at our biggest auditorium, I don't know, 1,000 researchers or whatever. And he gave such an amazing talk. He was like a rock star, okay? It was so moving. And just the day before he received the Genius Award, not the Genius Award, the $3 million Russian-American Tycoons Award, I forgot the name, just the day before. And he showed pictures of the small kids that, basically paralyzed and dying in a few months after getting his treatments. They are walking around up and about like normal kids. It's like unbelievable, okay? And an audience of 1,000 otherwise die-hard conservative NIH researchers were standing and giving him a standing ovation. I felt, wow, I'm lucky to be in this profession and to try to contribute. If I'll ever just be able to contribute 100 for that, I'm happy. The deed is done. And these are private foundations. So that's why I'm here today, to try and help you.
Well, Eitan, thank you for that. I'll share that I actually just did an analysis of NIH funding of the particular rare cancer that we're interested in, which went from zero over several decades, just a simple goose egg. After a key discovery was made, actually by a scientist who at the time was funded by our foundation, it ticked up to about $1 million a year. It included a $10 million center grant on top of that to that particular scientist. This year, it's almost $3 million, and every single investigator, as far as I can tell, was funded first by the foundation. And I can tell you, we are far from alone. There are many foundations like that that have, helped open up an area. It partly speaks to the role of patients and caregivers in driving understanding as well as providing financial resources, and in telling their stories and actually providing some of the key medical clues to investigators. In some cases, it's an individual like David Gadashek, who's worked on Castleman's disease, who had the disease himself, was also a qualified MD scientist, and actually has written an autobiography called Finding My Cure. I think that's the exact title. So this is not a one-off story. There are a lot of folks involved in it, and I'm sure that many people on the webinar, whether they're involved with fibrolamellar or any other cancer, have been passionate about that. And it reflects the passion that folks like you three show also about either a rare cancer or the common cancers, the whole set. But this is a case where what the patients and caregivers want, what we want as scientists and physicians involved in it, just aligns beautifully. And when we work together, there's obviously increased hope. So thank you for your time tonight. Thank you for the inspiration of what you're doing, and we'll stay in touch and try to continue the discussion in ways that might lead to better experiments and better treatments.
Thank you, Mark.
Thank you.
Nice to meet you
Thank
all.
you, Mark.
Thank you, everyone.
Thank you, everyone.
Take care.